Hello all, this video is going to show how to analyze the DNA sequences from the producers in the Tiny Earth project. And these instructions can be found in detail on page 145 of the manual. It tells you to go to the BLAST website, so this belongs to the National Library of Medicine. And there are a number of BLAST options, and the one that you want to use is nucleotide BLAST. And when you go into this website, you see that the first field is going to be enter query sequence. And the query sequence is going to come from the sequence of the producer that was provided by Retrogen or whatever company has been used for the sequencing. It's probably going to come in a Word or text file. So let's say that this is the number, identification number of the colony, the producer. So this is the sequence. So you're just going to highlight the whole sequence and then you can copy it. And then you come to the enter query sequence and then you paste. There are other ways to add the sequence. If it's provided as a FASTA file, you can upload the file, etc. But for our purposes, we are just going to paste the text. You can also add a job title, but it's not really necessary. So the search set is the different databases that are available for comparison, and in this case we are going to stick with the rRNA, bacterial RNA database, and as you can see we are going to do 16S because that's the bacterial sequences, and we don't really have to do much else. But I do recommend that you click on this box that says show sequences in your window, because especially if you're analyzing multiple sequences, then instead of erasing your previous result, you will just have a new tab opening. So we are going to click on BLAST, and depending on the complexity of the sequence and the speed of your internet, then this was pretty high. And this is what you have, and you can look at these results in different ways, but let's look at the numbers. So the numbers on the right express different parameters related to the coverage, so the matching of the sequence to whatever sequences exist in the database. The e-value is going to express how close this matching is, and the smaller the number, the better. And as you can see, all these sequences in the beginning are 0. So considering this is a bacillus, this is a very common occurrence. Bacillus sequences are very similar to each other, so you have to make it clear to your students that the only thing that we can gather from 16S is the genus. The genus of the bacterium. The species is, we would need much more advanced method in order to figure them out, but of course they can look at some of the highest ranking ones and then look, for example, where they were found, and any bacillus that has a high matching that has been shown in the soil, and maybe it has been described as having antibiotic production, that may be a possible candidate. Other things that can be gathered, for example, here this is a graphic summary, and again, as you can see, pretty much all the sequences are extremely similar to the original, to the sequence in the database. But this is it, and again, you can do multiple searches using the BLAST interface. Just check the open a new page, and that way you will be able to keep them open and kind of double check what's going on. Thank you.